Hi, it's Mrs. Vance again, and we're ready to go on to the next part of the notes, part 3 of the notes for Unit 8, and that's about the process of translation. And we talked about previously that transcription was making a copy of the DNA, but now we have to translate that message from the language of bases, which are found in DNA and RNA, to the language of amino acids, which is what makes up proteins. Okay? So we use another molecule of RNA called transfer RNA, abbreviated lowercase t RNA, and basically the tRNA functions as a language interpreter. So it's basically going to convert or translate the language from the language of nucleotides to the language of amino acids. They do this by picking up appropriate amino acids and then using a special triplet of bases called an anticodon to match up to or the complementary codons in the messenger RNA on the surface of the ribosome. So this is a representation. This is how your book represents transfer RNA. It shows at one end of the RNA molecule, it has an amino acid attachment site that's specific for a certain amino acid, and at the other end there's an anticodon here that is complementary to the messenger RNA. So if the messenger RNA is AAA, then the anticodon is going to be UUU because U is complementary to A in RNA. The translation occurs on the surface of ribosomes. Ribosomes are made of two parts, a large subunit and a small subunit. And then each one of those parts is made of ribosomal RNA molecules and proteins that are put together in the nucleolus in the nucleus of the cell. These subunits are separate until they're making a protein, and they come together during that translation process. There are binding sites for both the messenger RNA and the transfer RNAs, and there's also an enzymatic function that occurs there that makes the peptide bond between the adjacent amino acids. So there are basically two binding sites here. There's the A site, that's the first one, and the P site, which is where the peptide bond is formed. There's also, a lot of times they'll show a third site called the E site, which is the exit site, basically when after the transfer RNA transfers its amino acid over to the peptide chain, then it basically is ready to exit the ribosome. So we'll see how that works. And so basically here's our first transfer RNA is going to attach here, the second one will come in there, the peptide bond is here, and that then basically when the peptide bond forms, that's going to transfer this chain over to here, then it's going to shift everything over to the left a little bit. The ribosome will shift to the right and move everything over. This one will be released and the next one will come into place. Let's watch the video now to see how it works, and then we'll talk a little bit more specifics.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So you see it happens pretty quickly, okay? So there are basically a couple of steps here in the translation process. The first part is called initiation, okay? And there are two steps that happen here. First of all, the messenger RNA attaches to the small ribosomal subunit, and the first transfer RNA binds to the messenger RNA at the start codon. Start codon is AUG, so the anticodon on the transfer RNA is going to be UAC, and the transfer RNA is also going to carry the amino acid methionine. Once that happens, the large subunit attaches to the small subunit, and that allows the entire functioning of the ribosome. And basically, that's the P site has the growing peptide chain, and the A site is available to receive the next transfer RNA when it comes in with the next amino acid. So here's the start of our genetic message. Here's our AUG. The end of it is here. That's going to be our stop codon. I believe this one's UAC, or UAG rather, and the cap and the tail are there. So this is the part of the messenger RNA that's going to be read or translated into protein. Okay, so our initiator is our first transfer RNA that attaches to the, matches up to the start codon there on the small subunit. Then the large subunit comes into place, leaving room for the next amino acid to come in. Once the initiation is finished, then the amino acids come in one by one to attach after the first one. And then the addition of amino acids to the chain, to the polypeptide, is called elongation. So initiation first, then elongation, okay? And then this continues to make the entire length of the molecule, however many amino acids long it is. Some proteins are very, very large, having several hundreds of amino acids in them. Others are shorter, and that continues until the termination stage. In the termination stage, the ribosome is going to reach the stop codon, and there's going to be a place for something called the release mechanism to attach, and then that's going to release the polypeptide, and the ribosome is going to separate into its two components again, and the messenger RNA is ready to be chopped up and recycled. Here we have there are some, especially in bacteria, there are some messenger RNA molecules that can be translated simultaneously. In fact, in bacteria, it can happen even while the RNA is still being produced from the DNA. In eukaryotes, this can also happen. This is called a polyribosome. Basically, if you've got a long strand of messenger RNA, several different ribosomes can attach and continue making multiple copies of this polypeptide as long as the cell needs it to do that. Depends on what it is that is needed. Here's the process again. So there's our transfer RNA in the P site. Here comes our next transfer RNA to the A site to recognize the codon recognition occurs there with the anticodon matching up to the codon. The peptide bond is going to form, and that's going to transfer the polypeptide over to the transfer RNA in the A site. The first transfer RNA is going to be released, and the second one's going to move over to the P site. That's called translocation, and then the next process is going to occur. So it continues on and on like this, transferring that over, making the peptide bonds, transferring the polypeptide over to the first one, releasing the previous one, and continues on until it reaches the stop codon.